Thank you for the kind introduction. I think I would like to begin by thanking Dr. Weil, Dr. Halagaber, Dr. Hania Amar for the kind invitation to be here. Let me tell you, I'm really excited to be here in Cairo. It's my first visit to African continent. I'm really enjoying my stay here. So thank you very much. So my lab has been interested in stem cell therapy for cardiac regeneration. So I think you all would agree with me that heart disease is one of the leading causes of death in today's world, and there is no permanent solution to this problem. But stem cell therapy has lately shown lots of potential in repairing and regenerating the injured or infarcted heart. And different types of stem cells are being considered or tested in clinical trials for cardiac repair. But Kaplan cells or bone marrow derived MSCs have received lots of attention lately because these cells are easy to isolate. There are no ethical issues involved because the source is always adult tissue. And they secrete, as we learned in the first lecture, they secrete lots of growth factors and angiogenic factors. And most importantly, they are considered to be immunoprivileged because these cells do not express cell surface immune antigen, major histocompatibility, class 2 molecule, and they secrete several immunosuppressive soluble factors, including indolamine dioxygenase, TGF-beta, prostaglandin E2, NO, and several other factors. So they can suppress the proliferation of cytotoxic T cells. Thank you. And they can promote the induction of Treg cells. This is the mechanism of MSC-mediated immune suppression. So due to these properties, bone marrow MSCs are being tested in animal studies and in clinical trials. In fact, in several small as well as large animal models, after transplantation, bone marrow MSCs were able to improve cardiac function. And based on the encouraging outcome of animal studies, for the last 10 to 15 years, several clinical trials have been conducted with autologous or recipient-derived mesenchymal stem cells or Kaplan cells. I should make this habit. The outcome of clinical trials was positive, but it wasn't as effective as the outcome of animal studies was. And several reasons were given to explain muted response of clinical trials. But age-related decrease in regenerative and proliferative capacity of autologous cells was the main issue. Because in animal studies, they were all conducted in young recipients, whereas patients enrolled in clinical trials were in their advanced age, because that's when heart disease at least takes place. So allogenic mesenchymal stem cells from young healthy donors are the ideal candidate cell type for cardiac regeneration. In animal studies, allogenic MSCs were able to improve heart function, promote angiogenesis in the infarct areas after transplantation. The outcome of initial clinical trials with allogenic MSCs was positive, and there were no significant side effects observed. But the beneficial effects were short-lived. Also, the long-term fate of transplanted cells in these studies was not determined. So we wanted to assess the long-term fate of transplanted allogenic MSCs in the heart after transplantation. We did this translational study. We transplanted both autologous and allogenic MSCs in a rat model of myocardial infarction. So as we can see here, after 1 week of transplantation, both autologous and allogenic MSCs were able to improve cardiac function. After 3 months, cardiac function started deteriorating in allogenic cell recipients. At 5 months, there was no difference found in cardiac function in allogenic cell recipients and in control group. And when we analyzed survival of transplanted cells in these hearts, after 1 week of transplantation, both autologous and allogenic cells were present in the heart. After 5 weeks, almost all the allogenic cells were rejected by host immune system, whereas autologous cells were still present in the heart. In a separate study, we detected anti-donor alloantibodies specifically against transplanted allogenic MSCs. And the outcome of our studies was later on confirmed by several other reports. Therefore, allogenic MSCs, yes, they are immunoprivileged, but the cells become immunogenic after transplantation in the heart. And there is a need for in-depth investigation of immune properties of allogenic MSCs after transplantation before we take these cells to the clinic for large-scale clinical trials. So with this, one of the areas of focus in my research program is to understand immunological behavior of MSCs after transplantation. And we study effect of ischemic environment or hypoxia on immunogenicity of MSCs. We are also interested in understanding the effect of stem cell differentiation. This is more related to pluripotent stem cells. Effect of differentiation on their immunogenicity. But today I'll be mostly talking about the effect of ischemic environment on immunogenicity of stem cells. Immunoprivilege of MSCs is preserved by several immunosuppressive soluble factors secreted by these cells. A PhD student in my laboratory, Nikita, she did a study. She checked the levels of soluble factors in MSCs before and after exposing these cells to hypoxic environment and found that the level of prostaglandin E2 decreased in MSCs after they were exposed to hypoxic environment. Prostaglandin E2 or PGE2 is a immunosuppressive soluble factor. It promotes immune suppression by decreasing the proliferation of cytotoxic T cells, by suppressing natural killer cells and B cells. PGE2 also suppresses maturation of dendritic cells. PGE2 is produced in the cells by sequential oxidation of arachidonic acid by two important enzymes, cyclooxygenase 2 or COX2 and prostaglandin synthase 1. So when we checked the level of these two enzymes, there was a significant decrease in COX2, mRNA and protein expression, and prostaglandin synthase 1 levels. Therefore, PGE2 synthesis decreases in MSCs under hypoxic environment. The question that we wanted to address at this stage was this decrease in PGE2 levels that we found, is it responsible for loss of immunoprivilege? And to address this, we conducted several in vitro experiments. We co-cultured stem cells from Wistar rats with leukocytes from Lewis rats and studied the effect of lymphocytes on stem cells in terms of cytotoxicity caused by lymphocytes towards stem cells. So as we can see here, the level of cytotoxicity increased in hypoxic cells, and PGE2 treatment decreased the level of cytotoxicity. Another mechanism of MSCs-mediated immune suppression is by decreasing the proliferation of cytotoxic T cells. In our co-culture experiments, we assessed lymphocyte proliferation. Normoxic cells were able to decrease lymphocyte proliferation, But after treatment with hypoxia, these cells were not able to affect lymphocyte proliferation. PG2 treated hypoxic cells were able to suppress proliferation of allogenic lymphocytes. So PG2 preserves immunoprivilege of allogenic MSCs, and the level of this soluble factor decreases once these cells are exposed to hypoxic environment. That leads to loss of immunoprivilege. We also found that treatment with prostaglandin E2 preserves immunoprivilege of MSCs. So as I mentioned in one of the previous slides, MSCs are immunoprivileged because these cells do not express cell surface immune antigen MHC2,